So this is Irfan Rahman, the chair of the lung aging group of the RCMB, and along with my colleague, Dr. Beata Kosmidar, she's co-chair of the Assembly on RCMB and lung aging group, interest group. And we have the second retreat actually this year. Today is November 24th, 12 noon Eastern Time. Welcome you all from all over the world. And we have several time zones we have to cover. So we are here for this webinar. We have two outstanding speakers, Dr. Rao and Dr. Deng. They are both pioneer in basic biology of lung aging to disease mechanisms, how the infection affects the lung aging. So this retreat, we have 30 minutes each for both the presentations, and we may have some questions after each presentation. And please put your question on the chat box or raise hands, and we'll be able to pick up those questions and we present to our speakers. And then there will be, after an hour, there will be another forum discussion among all the our members and the speakers and the panelists. Unfortunately, Dr. Mareka, another co-chair, won't be able to join today. She is in Germany, so but hopefully we'll be able to cover. With that, actually, a brief introduction. I'm going to introduce Dr. Beata, would you like to say something as a co-chair?
Thank you. I think you covered everything. Thank you to other people who just joined us for being here. And I would like to welcome our speakers.
Great. Thank you so much. So with that, I'd like to introduce Dr. Puroshothama Rao Tata. He is an associate professor at Duke University. He is an rising star, just an outstanding researcher. I came to know from his last Aspen conference and reading his papers on lung aging and lung stem cells and plasticity, regeneration. So really nice with respect to the disease mechanism, for example, pulmonary fibrosis, to some extent to airway obstruction. So we'd like to hear from him about what really newer aspects of lung aging based on basic mechanism. So he is an associated professor. He has been pioneer in this original research, works on lung and organoid models, and his research is funded by the NIH. You see a lot of great research and publication include several Nature science papers, and a lot of speaking, the talks in the different societies. And he's one of the important thing here. You may see that among our members, the young the emerging newer scientists or the emerging investigators and early stage investigators, to see how Dr. Rao maintained his research profile from PhD to till now. So we'd like to hear from him how this encompasses with lung research. So with that, I'd like to invite Dr. Puroshothama Rao Tata to take over the microphone, please. Thank you so much.
Thanks, Dr. Rahman and Dr. Kosminder for organizing this meeting and retreat and inviting us. I'm really glad to be here. This is my actually first time attending this group meeting, and I'm looking forward to continue my participation in these group retreats. Those who don't know me, my full name is Puroshothama Rao Tata, but I go by my last name Tata, so feel free to call me Tata. I'm at Duke in the Department of Cell Biology and Medicine, and also I direct the Duke Regeneration Center. So our lab, before I get to that, I have a couple of disclosures. Our lab is not necessarily that specifically focusing on aging, but many aspects of our work is directly related to aging and age-related diseases, particularly in the context of lung. As you can see from this nice illustration from Dr. Kalhan and his colleagues, there is a clear association between aging and many respiratory diseases. As you can see, there are prenatal exposures and early life exposures, And it appears that there is a tipping point in the midlife. I wish I know where that is so we can prepare for it, but somewhere in the midlife, the lung function seems to sharply decline, even in an ideal respiratory or ideal life, healthy condition, but especially together with some prenatal and early life exposures, that seems to even sharply decline. And in many chronic diseases are associated with this age-associated phenomena. So as I mentioned, our lab is not specifically looking into aging, but we are mainly focusing on cell biology of lung repair and regeneration. And today I'm going to talk about, mostly my talk is going to focus on that. And so, as I mentioned, so our lab has been interested in understanding the cells and cell dynamics in injury repair contexts and in different diseases. So we are really fascinated by different forms of cell types that exist in lungs. So this is just an illustration where you can see different types of cells, mostly here epithelial cells, as you can see, but there are some immune cells and there are fibroblasts. They exist in different shapes, different locations. They show different dynamics throughout the life within at homeostasis, but also during injury repair. So we have been fascinated, are interested in understanding their dynamics over time and different contexts. So with that, this is just a skeletal view. I'm sure you are all familiar with that, but just want to show we have this upper respiratory tract and lower respiratory tract. Within the lower respiratory tract, we have these airways. These are lined by this pseudostratified or simple columnar epithelial lining. And underneath that, there are different cell types, stromal cell types, including fibroblasts, vascular components, and some nerve fibers passing through. And not to forget the immune cells that are present within this tissue. And at the distal end of it, we have these gas exchanging unit, which we call alveoli or alveolus, where we have the epithelial lining and interstitial spaces occupied by various cell types, including fibroblasts, pericytes, microcapillary network, as well as many immune cells. So I'll briefly start with some of the work that we published in the last couple of years ago in focusing on airways, and then eventually move into the alveolar tissues. And so again, my talk will mainly focus on different cell types that are present in the lung and their dynamics, and also the models that we have been using and also developing to understand the cell biology of lung regeneration to get to the mechanisms. So again, my talk will focus on cells and models and mechanisms of lung injury repair. So as you all know, at the macro scale level, in humans, there is quite some heterogeneity, human lungs. We know there is a right lung, left lung, and within each of these lobes, we have subdivisions. I'm not going to get into all the details. And also in airways, there are many branches. And that can be nicely illustrated using this plastic casting by Dr. Weibel and his colleagues, where you can see airways are filled with this plastic casting. So we can illustrate the complexity and the branching pattern of the airways. So in humans, it's highly stereotyped. This can range from, depending on the age and body size, the number of branches can range from 23 to 25. That's the estimated, really experimentally proven. And within these airways, as you can see, so there is trachea, bronchioles, several generations of bronchioles. So now the question comes is, what are the different cell types that are present throughout these airways? Can we simply assume and say, oh, the cells present in trachea and mainstem bronchi and the very terminal airways are the same? We have a rough understanding of different cell types that are present, but we don't know the details, particularly a region that we have been fascinated by is called terminal and respiratory bronchioles. So as the name states, hopefully you can see my cursor on the right side, where there is the last branch of the airway that we call terminal airways, which is then connected to the respiratory bronchioles, which then lead into alveolar sacs. So the reason we got interested in this is there are many clinical studies, particularly radiographic imaging, where that came from Jim Hogg and his colleagues, where they have used high-resolution CT scan of both healthy as well as diseased lungs, particularly COPD and fibrosis, where you can see the terminal airway is nice and nicely organized in the healthy lungs, whereas in COPD, it appears that these terminal airways are either completely occluded or they show this peribronchial or fibrosis, whereas in fibrosis, like idiopathic pulmonary fibrosis, or in general in many interstitial lung diseases, where we see these terminal bronchiole-like structures are actually expanding. As you can see here, the so-called honeycomb patterning that we often talk about. So there is quite some dysregulation within these terminal and respiratory bronchioles. And this is even more important because, as you can see here, some clinical studies have actually shown that the disruption of distal airways actually is the initiating event in, and that correlates with the severity of COPD. So what I'm trying to say here is that, as you can see here, so in distal airways, there is a dysregulation even at the very early stages of COPD, like in GOLD stage 1, compared to, if we compare that with the parenchyma, where parenchymal changes are not happening until even late events, actually. So, which suggests that disruption of distal respiratory airways is present early in the disease, at least in the COPD, and that it precedes emphysematic changes that we see in the parenchyma. And now the question is why we didn't know much about these cells besides that they are associated with the diseases. So the main reason is that we have been using most of the groups in the lung field have been using mouse as a model system. And interestingly, mice doesn't have respiratory bronchioles. As you can see, this is the terminal airway that directly leads into alveolar ducts and alveolar sacs. Whereas in humans, we have this interspace structure, which we refer to as respiratory bronchioles. So these are absent in mice, and because of that, we didn't know much about what are the cells that are present. So to get to that, one can use single cell sequencing approaches these days, or even spatial transcriptomics, and that's exactly what we did. So if we simply take a piece of, or sample a piece of this lung, depending on where we pick, mostly we get parenchymal cells, not so much airways, particularly the terminal airways that are present, because those are extremely small. I'll show you later how small they are. And because of that, we kind of missed out some of the cells that are present within these extremely small regions. So to overcome this challenge, two former postdocs, now they moved on to start their own groups, took an interesting approach where we micro dissected these airways, as you can see. So we take such one of these lobes or secondary lobule, and then one can dissect out the airways from all the way from proximal to distal, and we can separate these based on the diameter. So that's what we did. So these upper ones are roughly about greater than 1 millimeter in diameter, and lower pieces are anything less than 1 millimeter in diameter of the airways, which can be shown here. So this is the one of the terminal airway, and that's connected to respiratory bronchioles and some of the alveolar sacs that are associated with the tissue. And as you can see, this is the real scale, where you can see the diameter of this airway is roughly about 250 microns. So these are extremely small. And as I mentioned earlier, so getting such small regions from simple bulk preps of any region may not give you representative number of cells, actually. And using these pieces, we have performed single cell transcriptome analysis. We also combined that with some of the old methods that are at that time available, Visium from 10x Genomics. So combining those data sets, we found the different cell types that are present. I'm not going to go through all the listed ones here, but the ones that are listed in these gray boxes are the ones that belongs to the terminal and respiratory bronchiolar regions. So again, in a schematic view, so this is the terminal and respiratory bronchiolar region, where we found these yellow cells as well as another blue cell that's also present within the alveolar sacs around the respiratory bronchioles, we call these AT0 cells. And the yellow cells, we call them terminal and respiratory bronchial or secretory cells. And these cells are also referred to as RAS by others, Ed Morrissey, Maria Basil and Ed Morrissey's group. So we published this work. They came together back to back at the same time. So now, again, I'm not going to go through all the details. It's already published. But what we found is that some of these cells are either expanding. For example, in COPD, we see an expansion of these we call these AT0 cells. So these, some of the characteristic markers are SCGB3A2 and SFTPC. These co-expressing these cells that mark these the AT0 cells. But also in fibrotic lungs, what we found is that either AT2 cells or the AT0 cells can actually make these TRBSCs. And that's where we think that there is a de novo emergence of bronchiolization, meaning alveolar epithelial lining is turning into more like a bronchiolar epithelial lining, which we demonstrated using organoid models. And that's all published. Again, I'm not showing that data. So, which indicates that plasticity within this zone, within the terminal and respiratory bronchioles, as well as some of the alveolar epithelial cells, particularly type 2 cells, contribute to some of the pathogenesis that's associated with both COPD and pulmonary fibrosis. So now we are continuing this. We are using other model systems, particularly ferrets. Together with John Engelhardt's group, we are continuing analyzing these regions through lineage tracing studies. Hopefully, I can share that in the future. And not only our ongoing work, but more recent papers from Hackett Group as well as Renat Shekiv's group and Maur Soler and Ivan Rosas, Naftali Kaminsky's group also actually described now similar cell states, not all of them, but some of them in different diseases, particularly in COPD and pulmonary fibrotic lungs. Again, implicating these cell states and their dysregulation in a variety of diseases. Now, moving on to the parenchyma, which is the alveolar part mostly. So as you all know, so there are airways and they lead into these alveolar sacs. So there is basically functionally two compartments. We have gas conducting units and then gas exchanging units. And within the gas exchanging units, we have these tiny alveolar sacs, which are lined by alveolar epithelium, which is extremely thin, particularly one of the cell type. We all know the AT1 cells, and then there are these AT2 cells, which are generally often referred to as cuboidal, and also they serve as stem cells based on some of the lineage tracing, their contribution to repair. They can self-renew and differentiate into type 1 cells. So one question we have been fascinated by is how are these type 1 cells, type 2 cells, the small cuboidal cells, able to generate these large flat type 1 cells? And to understand that and their interaction with other cell types, what we do is we look at each tissue type. When I say tissue types here now, epithelial tissues or capillaries, vasculature, and stromal cells, particularly alveolar interstitial fibroblasts, and not to forget the extracellular matrix that's present within these interstitial spaces. How do they work together to facilitate repair and regeneration when there is damage? But also, what happens to these components in different diseases has been a major question in the field as well as in our lab. So the rest of my talk, I'm going to mainly stress on two major things. What are the cell types that are responsible for sensing injury and initiating repair? But also, what controls tissue homeostasis? And to get to that, we have some injury models already. So one injury model that we've been recently using is called BHT injury, butyl hydroxytoluene. It's a food preservative. If you look into any of some of the preserved food that you come across, most likely they have BHT actually. So what I'm showing you here is in a control lung, alveolar type 2 cells are lineage labeled. That's why you see this TD tomato in type 2 cells. But when we damage them, we can see that these type 2 cells start to undergo some morphological changes. And that morphological change is also associated with broader tissue remodeling, as you can see, day 6 and day 10, and eventually they go back to normal. Yeah. And as you can see, these lineage labeled type 2 cells, small cuboidal cells, now stretched out into generate some type 1 cells, or some remained as type 2 cells. So the question is, what's happening during these intermediate states during the injury repair process? And this is also shown here histologically in control and injury in between, and finally they come back to normal states. And so we have been interested in this process, and to understand this, we need to develop more precise models. And that's where a former postdoc developed an epithelial organoid model where we can take type 2 cells and expand them in large quantities and keep them as either type 2 cells or differentiate them into type 1 cells, and they can go on to make these large balloon-shaped alveolus-like structures. So we call it a serum-free, feeder-free, and we have an expansion medium and differentiation medium. And using these tools together with the in vivo models, again, two former postdocs, Yoshi and Alex, have done single-cell transcriptome analysis of both in vivo as well as ex vivo models, where we can now see there are type 2 cells, type 1 cells as expected. Some other type 2 cells become activated after damage, and then some replicate and some go on to differentiate. And during this process, we see these emergence of these intermediate cells or the transitional states. Now, there are a couple of groups described this. So we call them PATs, pre-AT1 transitional states. Other groups call them keratin 8 high ADI, DATP. Again, many groups have now identified these cells that we think are coming during the differentiation of AT2 cells into AT1 cells. So you may ask, like, why this state is important or why we need to look into them. So it turns out that during these transition states, many signaling pathways emerge, and they are very relevant to many lung diseases, including emphysema, generally COPD, but also pulmonary fibrosis and many, many other diseases that have been reported recently. And one of that pathway is P53. So P53 is one of the highly studied protein probably. And it has been studied more in the context of tumorigenesis, and we don't know really much about that in the alveolar epithelium, and that's where we deleted P53 to see what happens during this process. Again, P53 only transiently comes up, but then it's lost once the type 1 cells are made. And so just to visualize what a phenotype looks like, so this is a schematic, a 3D visualization of alveolar sac, where you can see green alveolar lining, but also lineage labeled type 2 cell upon damage. This is an bleomycin injury where it differentiates into this large flat type 1 cell. Whereas in P53 knockouts, what we see is that these cells become large, but they don't become the true type 1 cells. Instead, they bulge out, actually. And the analogy I like to put it is, in controls, it's more like a thin crust pizza, whereas in P53 knockout, it's more like sourdough, the same material, but essentially now, instead of flattening, it's just expanding. And that's where it turns out that P53 actually directly regulates a lot of microtubule genes. And as you can see here, so this is a single type 1 cell and its microtubule organization. Yeah. And if we knock out P53, this is completely disrupted. Again, with the interest of time, I'm not showing all the data, but we now proposed a model where normally microtubules are just dispersed all around the cell, but in this case, P53 tau, tau like a tauopathies, it's also called MAPT. Together, these two proteins regulate bundling of microtubules, generate such a organization. And it turns out that there are also some risk variants in tau that are linked to both the COPD as well as pulmonary fibrosis. Yeah, which I think is an interesting area because this is obviously connected to aging. I think we need to, we have some candidates and that links pulmonary diseases to the aging. And I think these would serve as really good models to understand the disease pathogenesis. So just to summarize what I've shown you so far, when there is damage to the alveoli, these type 2 cells make these transitional states, which have very unique transcriptional programs. Normally they go on to make type 1 cells. However, in some diseases like pulmonary fibrosis, we found that these transition states just simply persist for some time, which is then associated with pulmonary fibrosis. And that's where now we have a link where likely the disease initiating events are happening in the epithelium, which then dysregulates the niche, underlying niche, particularly fibroblasts, which then becomes myofibroblasts. So two former lab members wanted to understand what's happening in this context. So what they have done is they now looked at the mesenchymal cells. So we performed single cell transcriptome analysis of BHD injury, but also AT1 ablation, which I'll show you later, and combined that with other existing data sets. And we came up with this plot just to simply show you there are normal fibroblasts, there are injury-induced or repair-associated fibroblast states based on some of these characteristic markers. So we like to call them, so normally alveolar fibroblasts, sorry. And then we see these alveolar fibroblasts transition to something we call primed or inflammatory fibroblasts, which then become remodeling fibroblasts, or the so-called myofibroblasts, or CTHRC1 positive fibroblasts. And so what's happening in these different cell states during injury repair? As you can see, if we look at the ECM profiles of all these cell types, we see quite an increase in these ECM scores, and many of them actually have been implicated in emphysema, particularly through GWAS studies, so which indicates that these proteins or genes are playing a role in some of these transitions within the fibroblasts that happens during injury repair. So the same thing if I show you, if this is a normal injury, a normal alveoli, when there is damage, we see emergence of inflammatory fibroblasts, which then transition to or induces epithelial transition state that I mentioned earlier, and then which then induces a remodeling fibroblasts, other words, the myofibroblasts. So this is the process that happens. However, in a normal context, this goes back to normal during injury resolution phase. However, if we disrupt any of these through genetic or environmental factors, what we see is that instead of these tissues, repairing tissues going back to normal, they actually accumulate these myofibroblasts as well as some of the transitional states in this context of like activation of P53, or if we induce more transition states or activate RUNX1. On the flip side, if we knock out some of these, what happens is tissue cannot actually rebuild the lost structure. Instead, it undergoes simplification, something we see in emphysematic tissues. In other words, what we see is that this kind of serves as like a two sides of the same coin. In a way, injury is the coin where it can flip either to a fibrotic event or to an emphysematic event, where different cell types either accumulating or underrepresented, which leads to dysregulation within the injury repair process. So I have some couple of more slides, but with the interest of time, I'm quickly skipping to a schematic where what I want to show you is that this during injury repair, the interaction between the epithelial cells, the transitional states, as well as these alveolar fibroblast transition states, where it's not just the homeostatic cells interaction only, but the interaction between the transitional states themselves within these alveolar epithelial as well as alveolar fibroblast components that we think is essential for orchestrating the proper injury repair process. Defects in either of these compartments, and I'm sure many other components like vascular and immune and various things are playing a role, but just from our work, what I can point is that the interaction between these two is really critical for proper injury repair process. Without that, that can lead to some of the injury states. And also, I didn't get a chance to show you the data, but one of the things I'd like to highlight is that in any of these events, if we think about what is the most functional cell type in the lung, is the alveolar type 1 cell, for the reason that it's important for the gas exchange purpose. We need to bring this back in all these injury models or disease contexts so that tissue can probably repair itself, revert from a pathogenic state to a normal tissue. So with that, I'd like to briefly acknowledge all the current lab members and some of the past lab members and many collaborators. Without their help, we wouldn't be here. We wouldn't have been able to done this work, and many funding resources. And finally, thank you for joining this retreat. Thank you.
Thank you so much, Dr. Tata. Very fascinating. So now we have time for a couple of questions. And then remember, bear in mind that we'll have more discussion toward the end after the second speaker, Dr. Deng. So, if anything on chat you can put or raise hands, and Daniel will be able to pick up our ATS host and then communicate to us. So in the meantime, while we are thinking of question, Dr. Tata,
Sure.
first of all, thank you. I have a small question. Do you think any lung aging related aspects before the disease can happen in the transitional cells? Like one of the hallmark can be senescence or some of the changes. Is it possible? In other words, the TP53, if you remove that cell undergo senescence. So those are the two tiny questions. So please see whether you can address that. Or if you don't know, then that's fine.
No, absolutely. That's an area actually we are actually looking into. So hopefully I can answer your question. So this P53 activation, that state, also shows the hallmarks of senescence actually. So this is a transient event, meaning normally we don't see that senescence in type 2 cells or the type 1 cells, but only during that repair process, transiently these cells activate that program. Yeah. And if we activate that program, for example, we have done that by deleting MDM2 in type 2 cells. So if we delete MDM2, that activates P53, that in itself is sufficient to get this transition state, and which then induces fibrosis. Yeah. Yeah. And now I know many groups are actually interested in targeting this cell state, particularly the senescent state, to prevent the disease pathogenesis process. Yeah, I think it will be interesting to see in the coming years.
Excellent. Thank you. Are there any other questions and feedback to Dr. Tata? Yeah, there are some questions. I can see in question answer. There's a Jonathan Kurche asking, could you discuss the structural or pulmonary physiological consequences for microtubule interaction with TP53 in barotrauma or velotrauma? This is a hyperoxic condition. Yeah, in children.
Yeah, thank you. Thanks for that question, Jonathan. So currently what we know is that these type 1 cells have this really unique organization, particularly these microtubules, as I shown you earlier. And we see this is a culture cell, but even in vivo, we see such organization. Yeah. And how that changes with the different type of trauma is an interesting question. To address that, one thing we have to keep in mind is, any of these injuries should not damage the type 1 cell, for example, because if the type 1 cell is damaged, that whole organization is gone anyway, right? So we have to keep that in mind. But at the same time, this is somewhat connected, meaning, what are these microtubules actually doing in type 1 cells? Is that actually providing the structural support for this large, extremely thin cell type? And that's where we are currently looking into. What happens if we disrupt these microtubules? Does it alter does it break the type 1 cells? Right. That's an ongoing work. Yeah, but thanks for that question.
No, good. Thank you so much. So I don't see any more. Beata, you have any question, or Dr. Deng?
Yes, I would like it's a great talk, and I have one question. You mentioned both chronic and acute lung injury, and different factors can be involved here. I wonder if you have a chance to compare transitional epithelial stage and their restoration. What are the differences after chronic and acute lung injury, if any?
Yeah, absolutely. So in both conditions, both chronic and acute injuries, we see them. And in fact, actually, I didn't get a chance to show that data, but we actually simulated some level. Maybe this is the time I can show. Oops, looks like my screen unshared. Yeah, you still see my screen. So this is a control lung, no damage, and you see nice, beautiful alveolar lining. But this is AT1 cell ablation. So here we are actually testing, specifically ablating the type 1 cells. And what we found is there is just, so in this case, there is no bleomycin, no flu injury, nothing else, just eliminating type 1 cells only. If we do that less than 40%, what we see is there is tissue-wide, organ-wide remodeling, and these lungs go back. And during these phases, we see nicely the transition states, both in the epithelium as well as fibroblasts come up. But here is the thing. If we do that ablation beyond 70%, so we can dial this because we have this conditional, this ablation is using tamoxifen base, so we can increase, decrease the tamoxifen levels and thereby regulate AT1 ablation either below 40 or above 70. And when we did this, what we found is these tissues undergo simplification and they don't revert. Yeah. And this is where we see that the tissue is no longer making any more transition states, actually. Yeah, when there is extensive damage. Yeah, and to directly address your question, we have also done this 40 or 50% ablation, multiple rounds to see whether this tissue can repair. And young animals can repair, but not the age. Yeah, so it's not published yet, but we're still currently working on it. And I think these models can really give us some insights into fine details about what are the cells that specifically are controlling or surveilling tissue organization, and without which there is remodeling. And in some cases it can go back, but in other cases not, like aging conditions.
Thank you. So we have one more question, the small question, Dr. Tata, for you. And this
Yeah.
is from Dr. Shah Hussain. And he's saying it's good to see you always. Great talk. He acknowledged you. And the question is, if the loss of TB occurs earlier than the alveolar damage, and Dr. Shah is wondering whether if you are looking at alveolar repair, and that will also affect TRB repair at the same time.
Yeah, I wish I had the answer for that. These are really some complex questions because, in mouse modeling, that is a bit tricky. We don't have the RB regions, and that's where we hope the ferrets are going to be really useful.
Correct. Yeah, that's why I say he's working on the injury model. So, final question, tiny question by Jonathan, saying that at what age the mouse does repaired injury stops? I mean, human versus mouse. So, just a brief, very tiny.
Yeah,
What
sure,
age?
sure. Yeah, we think the tipping point is around 18 months.
18 months. Got it. Thank you. Okay, so we conclude Dr. Tata's presentation, but he's around. We can have open up again the forum discussion if he can stay another 30 minutes. So I'd like to take my co-chair, Dr. Perda Kasmidar, professor at Temple, take over the microphone to introduce Dr. Jane Deng. Please.
Thank you. I'm pleased to introduce Dr. Jane Deng, a professor in the Division of Pulmonary and Critical Care Medicine at the University of Michigan Medical School, and she's also affiliated with the VA Ann Arbor healthcare system. Dr. Deng studies how influenza infections alter immune responses to secondary bacterial infections using both in vivo and in vitro models and clinical research specimens. She investigates the role of different neutrophil phenotypes in various infection contexts and aging. Additionally, her lab explores how changes in the microbiome or type 1 interferons caused by influenza infections influence immune responses against bacteria. Please join me in welcoming Dr. Deng.
Thank you very much. Thank you, Dr. Cosminder and Dr. Rahman, for inviting me. And Tata, that was a really fascinating talk. I learned a lot. So as a disclosure, I, like Tata, I don't have, my lab doesn't really specialize in aging, and I don't really have expertise in aging aside from my own personal experiences with aging. But I'm going to talk a little bit about what we have observed in aged hosts during viral infections. Let me see if I can. All right. And these are my disclosures. I don't have any commercial disclosures, and this is my funding sources, as well as I want to highlight our NIH T32 in support of our postdoc whose data I'll be presenting. My lab has recently been focusing predominantly on this problem, which is what happens during viral infections when you have a lower respiratory tract infection, especially during flu or COVID, and what kind of mediates resolution versus persistent neutrophilic inflammation that results in ARDS. And the one thing that we will note during all viral infections, especially flu and COVID, is that older hosts are more susceptible to severe viral infections. And so this is data from the most recent completed flu season 24-25, looking at influenza-related hospitalizations. And you'll see that as age increases, the rates of hospitalization goes markedly up, especially among the older than 65 age group is really when it starts to take off. And so to examine sort of underlying mechanisms underlying why we see this increased severity, my collaborator and colleague, Daniel Goldstein, who tragically passed away a year and a half ago, who's pictured here, his lab has really focused on the association of aging and severe influenza infection. And what his lab did was they intranasally administered this influenza strain to young, aged, and advanced aged animals. And aged animals, they defined as 16 to 18 months, and then the advanced age were 22 to 24 months. Young mice were 2 to 4 months in age. And just like what we see in human populations, aged animals had worse outcomes. In this case, you will see a stepwise progression where the young mice had pretty much this dose that they administered was under an LD20. And as age increased, survival progressively worsened. This was also associated with, actually, interestingly, pretty comparable weight loss, except maybe, the middle part of the acute infection phases, the advanced aged had perhaps a little bit more rapid weight loss compared to the young. And interestingly, this was not associated with necessarily significantly increased viral burden until we get into later stages at 9 days after infection. So one of the first early questions that he and I asked after that paper was published was whether or not the microbiome mediates impaired host defense. And this is a review paper that we published a while ago that kind of, you can, if you're interested in sort of the rationale behind microbiome and host defense against infections, you can look into more detail there. But this is basically what other people have observed is that if you administered antibiotics to a SPF mouse, you can see impairment in multiple aspects of host defense. And this leads to increased viral titers as well as increased bacterial loads following infection. There's also studies in human populations that the gut microbiota does change with age. And this is sort of like a principal components analysis of what the gut microbiome looks like. And it's believed that the gut microbiome will mediate, like I said, aspects of innate immunity. But in this case, in human populations, they saw that the young gut microbiome was very different than older, healthy, community-dwelling elderly people in the green dots. And then the sick and hospitalized patients were the red and yellow respectively. And that there was sort of this distinct cluster, and this was also associated with overall health in older populations. And then finally, there was a group that published that if you transfer aged gut microbiota to germ-free mice, that this was associated with the systemic inflammation that we see in older hosts. So this seemed to be a pretty strong rationale for pursuing this aspect. And so what we did was we decided to co-house young and aged mice that are, they're not really gray, these black 6 mice that are aged, but we just depicted it as gray. So we co-house these animals and then basically collected fecal samples and then infected them after 90 days, which published studies had shown was sufficient to basically have the animals kind of converge to a microbiome of their litter mates, because mice do eat poop, each other's poop, and that's how they get their microbiome. And so after 90 days, we infected them with this PR8 strain of influenza and then recorded outcomes. And disappointingly, we did not see that co-housing resulted in any sort of effect, either by survival, body weight, or by, really by clinical score. And I only mention this by way of saying that negative data, I believe, is worth talking about, because we certainly want to save other people from making the same mistakes, and hopefully can build upon each other's work. So if anyone has questions about this or wants to pursue microbiome studies in age, just please reach out to me. We're happy to share our experience. So conclusions from this microbiome experiment is that co-housing does not seem to improve outcomes following influenza infection when you co-house an aged mouse with a young mouse. Part of the problem may be that the duration may not have been sufficient. And so in the future, we'll need to confirm with microbiome analysis to see if microbial composition actually does change. And the other thing that we were thinking is that microbiome dysbiosis may be real in aged humans, but not well represented in our SPF animals, especially since most of our animals have been on a stable diet that's free of fast food, alcohol, and processed sugar, which is what we observe in aged humans. And finally, the microbiome effects may be dwarfed by other age-related immune dysfunction. So the next thing we looked at was whether or not enhanced inflammation causes age-related susceptibility to flu. We wanted to look specifically at neutrophils because human populations, we can see that if you die from ARDS, you have persistent neutrophilia in your lungs compared to people who had ARDS but then subsequently lived. And the thing I will highlight here is that it's the persistence of neutrophils and not necessarily how high your neutrophils were at the peak that really seems to govern outcomes eventually from ARDS. So in our aged animals, even though, like I said, the viral loads were pretty comparable at the early time points, the neutrophil numbers were markedly elevated in the aged mice. And this kind of persisted over time. And so what we wanted to do was deplete these neutrophils to see what role they played. And so we would administer this anti-Ly6G or 1A8 antibody to deplete neutrophils, and then at the same time that they were infected with flu, and then kind of look at various outcomes. And what we'll see here is that if we depleted neutrophils starting at day 0 in the young mice, this actually resulted in worsened survival. Whereas if we depleted neutrophils in aged mice, this actually did not change their lung injury, which was, or their survival, which was surprising to us. Conversely, though, if you depleted neutrophils starting at day 6, we did not see the same detriment in the young mice. However, we saw that this depletion was actually protective in the aged mice, suggesting that neutrophils in young and aged mice are fundamentally different. And not only that, over time, the role of these neutrophils are fundamentally different in young versus aged animals. What we'll note here is that the neutrophil depletion did not actually lead to impaired viral clearance at later time points. So this is day 10 after infection. But it did decrease lung injury both in the young as well as aged animals. Specifically, it also was associated with decreased inflammation, especially in the aged animals. And so we think that the benefit of neutrophil depletion in aged animals seems to be mediated predominantly through decreased lung injury. One thing that I do want to make sure folks understand, though, is that neutrophil depletion does play a role in viral clearance in young animals. And so if you deplete neutrophils in younger animals, you will see increases in viral titer. And this is associated with worse survival. So they do play fundamentally decreased roles. So the interim results from these studies are that neutrophils are robustly recruited to the lungs during influenza infection, but are higher and persist in age compared to young animals. Neutrophils contribute to viral clearance of flu strains in young mice, and so early neutrophil depletion can lead to more lung injury because of this increased viral load, and it can lead to worse survival. But in old mice, neutrophils contribute to lung injury without affecting viral clearance, and so therefore late neutrophil depletion leads to less lung injury. We then wanted to look at what mediates this prolonged neutrophilia in aged animals. And going back to the original study where Daniel's lab had observed that alveolar macrophages were decreased in older animals compared to young animals. And when they depleted alveolar macrophages using clodronate, they found that both in young and aged that survival was decreased following influenza infection. He also found that when you transfer young alveolar macrophages to aged animals, that you had decreased lung injury. And here, this is an LDH assay where they transfer young alveolar macrophage to advanced aged animals. They had decreased lung injury compared to aged animals that had got aged neutrophils. And then these are the controls that didn't get any macrophages at all. So these are all macrophage transfers. Notably, this did not affect viral load at all, having exogenous alveolar macrophage transfer. And then he also found that that neutrophil efferocytosis by these alveolar macrophages overall was decreased in advanced aged animals. And when you depleted alveolar macrophages in the aged animals, you had higher myeloperoxidase, which reflects increased neutrophilia. So the thinking here is that you have persistent neutrophils in older lungs following influenza because of decreased macrophage numbers in aged animals, which then presumably leads to decreased efferocytosis of these neutrophils. And I'll also note that Dr. Kosmadir has previously very kindly shared with us human data, which shows similar phenomenon in aged people, that they seem to have a trend towards decreased alveolar macrophage numbers, even during states of health. So the mechanism underlying this alveolar macrophage number decrease, we think, is mediated by PGE2. And so in this paper that was published a few years ago, we looked at the source of PGE2 in the lungs during states of health and found that both in young animals as well as aged animals, when you isolate alveolar epithelial cells, That seems to be the predominant source of prostaglandin E2, whereas alveolar macrophages really wasn't a major source of this prostaglandin. And then during viral infection, over time, you can see increasing levels of PGE2, especially in the aged animals later on compared to their younger counterparts. So what does PGE2 do in our model? So when we blocked PGE2, the receptor, which is EP2, as depicted in the purple dots, we can see that EP2 blockade leads to increased alveolar macrophage numbers, both at baseline as well as over time during influenza infection. And this increase in alveolar macrophage number is also associated with decreased markers of lung injury, in this case represented by albumin concentration in BAL fluid. And excitingly, this was also associated with improved survival compared to vehicle control. And as a control to see if there's actually two receptors for prostaglandin E2, EP2 and EP4, and we did not see any additional benefit with blocking both receptors compared to just EP2 alone. So to sum up this mechanism, what we believe happens during aging is that in young animals, we have type 2 alveolar epithelial cells that don't make PGE2, and through a mechanism that's likely mediated by mitophagy, which I will not get into today, this leads to improved alveolar macrophage health, which allows them to proliferate and increase in cell number under a healthy state. And then during infection, this promotes appropriate immunity against influenza infection, both decreasing inflammation that's mediated by neutrophils, as well as decreasing the viral load. Whereas in aged lungs, senescent type 2 alveolar epithelial cells make more PGE2. And this is believed to impair mitochondrial fitness in alveolar macrophages, which then leads to impaired proliferation and lower cell numbers, thereby increasing inflammation during aging. So finally, we wanted to take a translational approach and see if senolytics would then improve immune dysregulation. And I'm going to subtitle this A Cautionary Tale of 4 Senolytic Regimens. Given that this was sort of a relatively new area of my lab, we wanted to establish some of the procedures and techniques for looking at senescence in our animals. So one of the things we looked at was serum cytokines, inflammatory cytokines in healthy, young, and aged, and established that aged animals did indeed have increased levels of IL-6 and MCP1, as well as IL-10. We also looked at P21 levels in lung homogenates of old versus young mice, and then looked at beta-gal staining, and this is gonado-white adipose tissue, and found very nicely this assay worked, demonstrating that aged mice indeed had increased markers of senescence. And so the 4 senolytic regimens that we used where we first started with pre-treatment, where we gave basically 2 weeks worth of senolytics, and then had a 1-week washout period followed by influenza infection. And the 2 regimens we tested using this approach were dasatinib and quercetin, as well as fisetin. And then we later on tried the treatments concurrent with influenza infection. And here we gave again D and Q. And then at the suggestion of a reviewer, we gave navitoclax. And all of them failed miserably. And so the pre-treatment regimen where we gave D&Q or fisetin pre-treatment, no differences in body weight, absolutely no difference in survival after influenza infection in our aged mice. These are all aged mice that received these. And then when we subsequently pivoted to concurrent treatment with either navitoclax or D&Q, again, we saw no difference in outcome, either survival or any of the clinical scores. And so one of the things we were trying to understand is what was happening to our animals during infection. And so here you can see that D and Q did nicely decrease neutrophil numbers at day 8. It also decreased CD4 numbers at day 8 in the D and Q, whereas fisetin really didn't have much of an effect. Both treatments, both senolytic regimens also decrease NK cell numbers, and fisetin decreased B cells. But the net effect of all of these inflammatory changes was really no differences in lung injury. And I'm here showing the IgM in the BAL, which is a marker of lung injury, but I also have histology that there's really no difference in histology following treatments with senolytics. So then we wanted to ask the questions, well, were our senolytics actually biologically active? And so we tested these senolytics, dasatinib, quercetin, and fisetin on primary human bronchial epithelial cells and found that, yes, they did decrease phosphorylated SIRT1 with increasing doses. And we also saw AKT activation. And then finally, in our D and Q, we found that when we administered it in vivo, they did have a biologically active effects on lungs, with the D and Q treatment showing decreasing phosphorylated AKT. So despite that, however, we did not see any decrease in senescence markers in vivo. So although they had biological activity, they did not decrease senescence. So this is the gonado-white adipose tissue, where we, again, whether they were given vehicle or navitoclax, we did not see any effects on beta-gal staining, no decrease or no effect on P21 expression. And these are the sort of densitometry. So our conclusions overall are that age-associated susceptibility to influenza is associated with persistent neutrophilia and perhaps decreased NK T cell numbers. And there's observations made in human populations that elevated neutrophil to lymphocyte ratio is associated with more severe disease. Neutrophils of aged mice do not seem to contribute to viral killing, but do mediate lung injury. And decreases in alveolar macrophage numbers during aging is mediated by PGE2 produced by senescent alveolar epithelial cells, which promotes increased lung injury following influenza in aged animals. The microbiome, conversely, seems to play a relatively minor role in age-associated susceptibility. And our final take-home message is that senolytics need to be validated in your model. So do not assume that a senolytic is exerting its effects as a senolytic. But in our case, the senolytics didn't work. So acknowledgements, this is my lab. So Matthew Worley, our postdoc, and Cal Carver, our lab manager, contributed to a lot of the studies that I just discussed. And then Daniel's lab, this was a photo of his lab after he had had to step back. And Adrian Luna is a postdoc who did the work on the senolytics with the assistance of Kathleen, one of his lab technicians. So with that, I will take questions. Thank you.
Thank you. That was a great talk. And please use the chat box if you have any questions. And I see one of them now from Dr. Jonathan Kirsch. Could you conceptualize the neutrophil depletion results and enhancement of survival from lethal infection recently published by Ron Germain's group in Science? How do neutrophils promote clearance of non-bacterial pathogens?
Well, yeah, that's a really great question. I'm not familiar with that paper, so I haven't had a chance to see it. But that is something that we are trying to investigate, is whether neutrophils are directly responsible for viral clearance. And we are looking at this both in vitro or ex vivo. where we're taking isolated neutrophils, and we have studies from both human as well as mouse that neutrophils do seem to decrease flu, but it is very context specific and they have to be activated in a very specific way. The other thing I will also give you a caveat is that there's a lot of heterogeneity among the different mouse strains in terms of their ability to kill viruses. And we think this is reflected in human disease. And so I think that it's very mouse strain dependent in terms of how neutrophils kill virus. Yeah, so if you want to elaborate, I would be really interested in hearing more about what Ron Germain's group published.
Thank you. Dr. Rahman raised a hand.
Yeah. Dr. Deng,
Yeah.
excellent presentation, lot of good work, lot of good incitement about the senolytics and aging and infection. So my question can be a comment, or it's not a comment, it's just a question for asking your suggestion.
Yeah.
So do you think that the senolytic, I'm not surprised with your senolytics data. So with quercetin or the other ABT compounds or fisetin, do you think this is a thought process that pre-infection, if we give to older mice, will at least attenuate some of the neutrophilic inflammation, and or post-infection will have some sort of repair mechanism by, or at least delaying the process of injury in those post-treatment senolytics model. What do you think?
So your question is, do we see reduction in neutrophilic inflammation?
If we give if you give before infection.
Yeah. Okay, so do you mean, okay, so we did show that when we give senolytics before infection and then we give them flu, the D and Q did decrease neutrophil numbers.
Yeah. Okay.
The lungs themselves at baseline don't have actually a lot of neutrophils at baseline. So we didn't actually look at what senolytics did, in healthy states. So I don't know about lifelong, like if you were to start at age 50 for a human, if that would, because I just don't think that we have a lot of neutrophils at baseline in the lung tissue, even in older people.
Yeah. So in other words, I was trying to ask you, because
Yeah.
infection is, this viral infection or infection is such an aggressive model.
Yes.
And that might not reflect with senolytics because
Right.
senolytics are ETBT. They are just making life a little bit better. And
Right.
it's not removed.
Agree.
So that's what my question was.
Yeah, I completely agree. And I think that it's the same story with the microbiome. It's sort of like it probably helps, but it's not sufficient, like if you're really facing a truly pathogenic strain. So I agree with that. And I do also agree that we have to be smarter about actually modulating neutrophil activity, and making sure it's sort of like a just right sort of level of neutrophil function.
Yeah, thank you so much. By, at least by doing this experiment, we come to know something. So it always advancement of science. Thank you so much.
Yeah, that's why I like to show negative data. So, but
Yes.
I'm happy to share our experience. So again, if anyone wants to reach out to us, I'm happy to share with you the conditions we use, all the other things that didn't work. Because I do believe that, science, we're under an existential threat and we need to show the public more return on investment.
Yeah,
So
absolutely.
the more we can kind of share this information, I think, hopefully we can get closer to saving ourselves from the next pandemic.
Yes, thank you.
We have another question from Dr. Harish Narasimhan. Very interesting presentation. Thank you. At what level do you believe senescent 82-derived PGE2 is acting to impair alveolar macrophages? Does it make
Oh.
them more vulnerable to infection-mediated death, reduce cell renewal, or impair differentiation of monocytes?
So great question. Yeah, that's something that had Daniel lived and I think we would have written our next grant to look. Actually, the mechanism we were pursuing was the mitochondrial fitness part, and which probably does have play a role, obviously, in cell survival. And we do see that one of my colleagues did show that PGE2 does impair, macrophage self-renewal. I don't know about the differentiation aspect, but we do believe it plays a role in sort of like in situ self-renewal. So I think probably cell survival as well as self-renewal. And if I can find the reference, I will put it in the chat.
Thank you. And the second question from Dr. Jonathan Kurs. Clonal hematopoiesis with aging seems to skew toward a more granulocytic differentiation in aged individuals. Could this influence outcomes in aged mice?
I know that clonal hematopoiesis, definitely that has been brought up before. I have to confess, I don't actually know too much about how much the mouse process of clonal hematopoiesis reflects human clonal hematopoiesis. I don't know if you know more about this topic. Jonathan, please share or please unmute yourself and share your knowledge. I'd be interested in hearing about it, both about this and Jermaine's work.
If I can just chip in, the chip mutations, right? The clonal hematopoiesis. So basically mutations in hematopoietic system, cells in the hematopoietic system that can actually impact on systemic level. For example, if there is a gene like TET mutations are very common in chip mutations, and DNMT, these are like DNA methylation, chromatin regulation, very often associated. And there are a couple of papers actually implicating their role in COPD and various other.
Yeah, I guess I'm just curious, like, how closely does the mouse data align with what is seen in human clonal hematopoiesis? That's what I was.
I see.
Yeah.
I know there are a couple of groups actually generated these TET mutations, the same mutations, DNMTs, and looking into both COPD as well as in IPF. Seems like some of them actually recapitulate. Yeah. Again, I haven't followed fully, but.
Yeah. Because I just know like some of the aspects of like neutrophil development. I feel like in mouse and human, there are some differences. And so I'm always a little bit cautious when I approach aspects of granulopoiesis, right? Because like first of all, there are fewer circulating mouse neutrophils than there are in humans. And I'm not sure why that's the case, just systemically. They just have fewer percentage of neutrophils in their blood in mice compared to humans, like half the percentage. And the other thing is that their neutrophil armamentarium is a little bit different in mouse and humans. Like they don't have the same sort of granules. And so, so I'm always like, just cautious when approaching, but yeah, that's really, I think it's, very compelling sort of mechanism. It has come up in study section before. So I don't.
Actually, if I may ask one question, that's kind of follow-up of one of the earlier question by Harish. So the interaction between the neutrophils, the type 2 cells, and the macrophages, do you think the neutrophils are at the apex of that interaction, or is there a interconnected interaction between these? I'm wondering, like, who is initiating that?
Oh boy. Okay, so I think what happens is that let's just put unhealthy aging. Okay. So during unhealthy aging, your lungs or your airways or, alveolar epithelial cells become senescent. And probably over time, what happens is that they do make more PGE2. And this then leads to decreases in alveolar macrophage, and then neutrophil inflammation is sort of like the last sort of step. And I think that's normally, in my mind, that would make, probably the most sense. And the reason I say that is because we have shown that we can, if we make alveolar epithelial cells senescent, by UV radiation. Now, granted, it's not the same as aging-associated senescence. We can induce PGE2 production by these type 2 alveolar epithelial cells without any other cells in the system. Now, what is the mechanism of induction of PGE2 during senescence? I am not sure. We haven't really pursued that. And I'd be curious if other folks have looked at induction of senescence. The other thing that to me is very striking is that senescence is associated with activation of so many other molecules, right? And so including activation of NF-kappa B mediated cytokines. And so I wonder if, I just don't know, which, how much of a vicious cycle there is, like a feedback loop.
Great, thank you.
Yeah.
And I have one question. At the beginning of your presentation, you showed the neutrophil and macrophage alterations at different time points. I wonder if you had a chance to look at lung epithelial cells at these different time points in both young and aged animals, because it looks like there is a lot of changes.
Okay. Do you mean what aspects of lung epithelial cells?
The injury, recovery, or transitional stage.
Oh, yeah. Right. I have not definitely have not looked at transitional state. We've only looked at lung injury, but we haven't looked, we're not really an alveolar epithelial cell lab. And I would love to collaborate, because this is something that Rachel Siemens and I have talked about a lot, but we have not really looked at, the lung epithelial cells in terms of what's happening to them. Like, are they apoptosing or dying? Are the virus directly infecting them? Yeah, in great detail, or sort of like what sort of, processes are happening to them. And I think it would be very interesting to look at, especially in the context of neutrophil depletion, which I still hold is a central component of this lung injury.
Thank you. And we have two comments from Dr. Jonathan Kirsch in the chat box. Mouse models of TET2 mutations skew granulosity. And as far as the German group data go, they showed early 1-DPI at high concentration reduced damage in bulb C and B6. Later reductions were not helpful in their hands.
This is neutrophil depletion, right? I guess. Yeah, I'll have to look at that because yeah, I'll have to look at that. Thank you for sharing that with me, because really in our hands, when we've done neutrophil depletion early during infection in young mice, this has always led to sort of like equivocal effects on lung injury because of increased viral loads. So as far as I know, the neutrophils are important for clearing viruses in young mice, but less so in aged mice.
Thank you.
Yeah, but I'll look it up. Thank you.
Any other questions from the audience? I think we can move to the next point of our webinar. Dr. Rahman has additional points to discuss, which are of high interest to our audience and the lung aging group.
Okay, thank you so much for Dr. Tata and Dr. Jane Deng. for a wonderful webinar. So we have at least 5 to 10 minutes to discuss general aspects of this webinar. And then I will conclude the webinar by stating few of the lung aging missions from the ATS RCMB. So number one thing for the forum discussion would be the areas of lung aging in context of cellular plasticity. That can be Dr. Tata or Dr. Deng. What are those cellular plasticity which we can study in lung aging in context with developmental biology or diseases? Number 2 point is, what are those mechanisms of the lung aging? Can be mitochondria, can be senescence, can be any other thing, or epigenetic. We can target using emerging models and therapeutics. So point number 2 might be difficult, but point number 1 today we address. So I would like to just maybe ask Dr. Tata to tell us a little bit what he thinks, some key points from the perspectives on cellular plasticity to study lung aging in context with diseases. Please.
Yeah, sure. Thanks, Dr. Rahman. I think the more and more we see is, at least in animal models, that's where we can experimentally, longitudinally follow these models, where we see there is tremendous plasticity within the lung. We have airway cells, we have alveolar cells, both in the epithelium, fibroblasts. There is quite some plasticity where, one compartment is generating other, and vice versa. However, not all plasticity is good in a way. For example, in influenza injury models, where we see loss of parenchyma, alveolar lining, and then we see exodus of these cells from the airways. They just migrate and populate these regions. And now the question is, we see plasticity, but is that really good? In other words, when airway cells migrate to the alveoli, are they actually inhibiting alveolar repair? Instead, these airways are populating. Is that something happening in ILDs, for example, right? These migrated cells. And, in many ways, some of these events are inevitable because right after injury, these are just migrating. The question is, can we convert them to the beneficial cells? Right, these migrated airway cells, can we convert them into alveolar epithelial cells, alveolar fibroblasts, so that we have the right cell types at the right location for the proper functions? I think that's an area we have to think about. There are a couple of papers that came out. There is a paper from Ed Morris's group recently in Science, where they showed that the notch signaling between these airway derived cells is blocking alveolar repair. So identifying such signals and blocking them so that we can promote the alveolar repair. And how that turns out in a aging context is still to be, I think, in my view, I haven't seen someone really probing that, but I think that's something we need to look into. But in the there are we see actually quite some differences between mouse and human, particularly some of the regions I mentioned, and then there are other cell types we see. So I think we need to understand what is their role, especially at the terminal and respiratory bronchiolar regions. In a way, they are at the junction, right? If there is damage in the alveoli, what do these cells do? Versus when there is damage in the airways. And that's the site, as you can imagine, chronic bronchitis, for example, right? We don't normally see mucus cells in those regions, but now suddenly these mucus cells come up. So where do they come up? Where do they come from? Are pre-existing some of these TRBSCs, the secretory cells, turning into mucus cells? Or is there a migrated proximal airway cells occupying these niches and now generating mucus cells, right? Pathologists often call the proximalization, right, in these diseases. So what is that actual proximalization? The de novo conversion of the existing cells, or the proximal derived cells contributing? I think that, with respect to plasticity, that's something we need to understand so that we can either block their migration, maybe, right? Or promote the local cells to repair themselves. That's my take.
Great. Thank you so much for our junior emerging investigator. They look forward for this important research. So the second thing is just a brief. Before that, actually, I have my co-chair joined, Dr. Marika Lim, and I met her. I do have a picture, a fond memory of her, and she may want to introduce herself before I have move on to second point. For then we have 4 minutes to conclude. So Marika, just introduce briefly, please.
Right. My name is Mareike Lehmann. I'm very sorry for being late. I had an important meeting to take, but I'm happy to be with you. Now I'm a professor for translational inflammation research in Germany, both in Marburg as well as in Munich, and I'm interested in aging phenomena, specifically in cellular senescence, in COPD, IPF, and also lung infections.
Thank you so much. So I have second point, then we'll conclude this webinar. The second point is with respect to, lung infection or injuries, and the ARDS kind of phenomena or exacerbation. For example, I give an example. Whenever we have an exacerbation of lung disease, for example, COPD, either bacterial or virus, there's a, lung function declines and we don't know whether the lung become aged at that time. So, and what are the models we can use? What are therapeutic? But Dr. Jane Deng models really told us that there are certain things to avoid, certain things to do. So, what's the take home message, Dr. Jane, for our junior investigators to look for research those kind of things, please, for a minute or two?
Wow. To be honest, I think it's going to be very challenging to undo a lifetime of sort of, suboptimal habits. I don't think there's a magic bullet. Like none of these treatments are going to be sort of like a cure-all for everything unless you take them chronically and chronically make, the changes. Because I think, what we're all doing is we're all seeing a small aspect of aging. We're all focusing on one little piece, but really they're all integrated, right? Like what Tata's looking at with lung regeneration is intimately integrated with, prolonged inflammation, and we really can't uncouple all these mechanisms. So I think it's going to be very challenging to find one thing that really, is going to be the magic mechanism. They all kind of have to, I don't know. Yeah, just my experience with the senolytics has been very very educational in that regard.
Thank you so much. So with this, we conclude this webinar, and thanks to our speaker, Dr. Tata and Dr. Deng, and our panelists and co-chair, Dr. Beata Kosmidar and Maria Kalem. So I would like to just take a minute or so to tell about the plan. So here are the plan. ATS meeting, we look forward in Orlando, and we have a lot of K99R00 applications coming. So please do focus on those junior emerging investigators. Think about the RFAs. Lung aging NIH perspective is great. Look forward. Then look for AFR announcements. ATS application also, you can look COPD, INET, IPF, similar lung aging forum. We are thinking, please do come forward. If you are interested, we would like to launch this lung aging forum at some point. Membership activities, we would like to hear your opinion in our next meeting. Please reach out to us. You can take a screenshot here and please reach out to us. With that, I would like to take this opportunity to thank all the speakers, our panelists, co-chairs, and our audience and members for wonderful annual retreat. With that, happy Thanksgiving.